And today I'm going to talk about rapid adaptation to climate change in copepods and how that might influence physiological plasticity. And so I think everyone knows that the climate is rapidly changing. Temperatures are increasing, the oceans are becoming more acidic, and extreme events are becoming more common. And there are two main ways that populations are going to respond to this, adaptation and plasticity. And these two processes, of course, are not independent of each other. They can both inhibit and facilitate one another. And I think it's really important when we consider how populations might respond to changing climate to consider both ends of the spectrum. And so specifically today, I'm going to talk about, first, do populations have the capacity to adapt to climate change? And secondly, if they do, what are the costs of this plasticity, or of this adaptation? Specifically thinking about plasticity. And the goal here is to really not only understand what will happen to populations in the short term, but what might be the consequences to resilience when other stressors are encountered. And so we're using the Carbonic as a model for rapid adaptation. This species of copepod is a worldwide estuarine distribution. It's super abundant, it's a main prey item of fish, and because of this, it plays a really important role in global biogeochemical cycles. For our purposes, it's great though because it inhabits these really variable environments, both spatially and temporally. So for example, here is variation in pH and temperature from a site in Long Island Sound over the course of a year. And you can see there's really extreme fluctuations in both of these parameters through time. And so what this means in conjunction with the spatial variation in these two parameters as well, is that there's likely high genetic diversity for tolerance to these traits and high plasticity for tolerance to these traits. And so we can leverage these in a system that's likely very resilient to climate change. And so we use experimental evolution and reciprocal transplants to understand how a Carbonic will respond to changes in pH and temperature. So we collected individuals from the field in the Long Island Sound, we brought them into the lab and split them into two treatments. The control treatment, we're considering ambient AA, or ambient CO2 ambient temperature, and high temperature, high CO2 greenhouse condition, which we'll refer to as HH. And here, shape is going to indicate line. So circles are ambient control, triangles are greenhouse condition, and color will be the treatment that they're in. Blue, ambient, red, greenhouse. So we evolved these in the lab for 20 generations, and after 20 generations, we reciprocally transplanted them into the opposite environment. So ambient, greenhouse, greenhouse into ambient. And we ran this for 3 generations. So for each of these lines, we have an adapted and a transplanted condition. We had more than 3,000 individuals per replicate, 4 treatments per, or 4 replicates per treatment. If you do the math for the 3 generations, 48 samples total. And we used pooled RNA-seq at each of the 3 generations to quantify both gene expression and allele variation. So the first thing to note here is Pca, the genetic variation in F1. You'll see there's really strong differentiation across PC1, where on the left, we have strong clustering of the ambient line, on the right, the greenhouse line. And so we're clustering by shape here instead of color. And so this suggests that there's been really consistent shifts in allele frequency in response to selection after our 20 generations. And this pattern persists for F2 and F3 generations, so this signal of adaptation is present for all 3. But you'll notice that by F3 generation, we're starting to see a signal of treatment here, where the greenhouse line ambient conditions is starting to segregate along principal component 2. For the gene expression, again, we're segregating largely by shape here. So we have ambient lines and greenhouse lines. At F2, everything's a mess. There's kind of no signal that I can tell. But by F3, we've again transitioned to this treatment signal condition. So we're again clustering by color instead of shape. So what this suggests is that we have a pretty strong adaptation after these 20 generations in F1 for both gene expression and allele frequency, but by F3, this is beginning to converge on a treatment effect. And this especially is present in our ambient line, or greenhouse So we wanted to look more closely at the signal. To do that, we used discriminant analysis of principal components. So here we're trying to maximize between group differences and minimize within group variation. And so we build discriminant function space with our non-transplanted lines, and we fit those to this plot here. And of course, we're sort of requiring that they're going to be segregated on either ends of the spectrum. But then we can ask, where do the transplanted lines fall in discriminant function space? And so if we plot the greenhouse line in ambient conditions, we see that it moves towards the ambient treatment to some extent, and we can plot the distance that they move with this arrow. We can do the same for the ambient line in greenhouse conditions, and plot the distance they move. We do this for all three generations, and we see this really nice transition across the three generations where we're transitioning from a selective history effect here, so clustering by line, to a treatment effect here. So again, clustering by color instead of shape, indicating that expression is really moving towards the physiological optimum across generations. We do the same analysis for the allele frequency data, and at F1 and F2, we largely see similar shifts for our two lines. But by F3, the shift in allele frequencies is larger for the greenhouse line in ambient than for the ambient line in greenhouse. And you'll notice too that our greenhouse line is falling outside the distribution of all of the non-transplanted samples at each F1, F2, and F3, while the ambient lines are primarily within this distribution. So this suggests that there's been adaptive evolution across the generations. And so if we take these together, we have this shift in allele frequencies by F3 that's really pronounced. And I think that there's two different mechanisms that are likely driving this. The first is likely plasticity in our ambient line, where we don't see a shift in allele frequencies in any of the generations, but we see this shift in gene expression. So this is likely plasticity. But for our HH line, our greenhouse line, this shift is likely caused by a change in allele frequencies and evolution. And I should note that we've analyzed these data in a bunch of different ways, and no matter how we cut them, this signal is very pronounced. For shift in allele frequencies in the greenhouse line, it's not present in the ambient line. And I'd be happy to talk to anyone about these other analyses if you're interested. We next compared sort of the relationship between the changes in gene expression and the changes in allele frequencies. So this plot is the contributions to the DAPC from the previous slides. And you see there's basically no relationship between the two parameters. This, first of all, indicates that there's not any bias in calling SNPs from our RNA-seq. This is great from sort of a technical perspective. This allele-specific expression isn't affecting these results. But perhaps more biologically relevant is that there's likely few or little evidence for cis-regulatory variation. There could be two reasons for this. First is that we're sequencing from expressed mRNA. So if LD is weak, we're missing essentially any signal selection in promoter regions, which is likely where most of the signal would be. The second is that trans-regulatory factors may be under selection. This actually seems very likely when we look at the enrichment, the functional enrichment of our allele frequency data. So one of the main factors that come out here is regulation of gene expression. We see lots of enriched function here. This indicates that trans factors likely are under selection and likely are driving this divergence in gene expression, which is really nice to see. We also see functions related to reproductive success and growth and development in this category. For the gene expression data, we see a lot of interesting mechanisms related to metabolic and catabolic processes, ion transport, and homeostasis. So things that always sort of come out in greenhouse exposure in marine populations. One particularly interesting gene is carbonic anhydrase. This plot here is a gene expression in each of our conditions, where color indicates the generation. White is F1, gray is F2, black is F3. Carbonic anhydrase catalyzes the conversion of CO2 to bicarbonate, so it's really important. And we see much higher expression in our greenhouse line greenhouse conditions, and our ambient line, and our transplanted lines are showing some very interesting patterns as well of sort of increased gene expression after transplant. So this is an interesting candidate gene that certainly deserves follow-up. We next looked at genetic diversity across the 3 generations for all of the lines. So again, color here indicates generation, white F1, gray F2, black F3. And for our 2 non-transplanted lines, they're able to maintain genetic diversity fairly well across the 3 generations. The same goes for our ambient line in greenhouse conditions. But the greenhouse line in ambient does show this drop off at F3. And if you remember, that sort of corresponds to the same generation that showed the largest shift in allele frequencies. So this indicates that this selective shift in allele frequencies is resulting in a population bottleneck. And that may have negative consequences, of course, in the future. Okay, so to sort of start to wrap things up a little bit, do populations have the capacity to adapt to climate change? I think the answer here is yes. However, this comes at the cost of maintaining plasticity and maintaining genetic diversity. So this adaptation to greenhouse conditions drives this loss of plasticity. And so when we move these populations back into these really benign conditions, I think that's an important sort of thing to remember here, that we're not further stressing them, at least you might think. It still requires sort of this adaptive evolution to respond. This drives this reduction in genetic diversity, and I think it's reasonable to think that might further reduce plasticity. And taken together, I think that these multi-generation reciprocal transplants are really necessary for this sort of experiment. So had we just done a single generation of a transplant, we would have never seen the signal through time. And I think the signal across the generations is really what's informative about how populations might respond to further stress in the future. And I want to note, too, that this experiment is part of a much larger selection experiment that we're conducting right now. So we're doing a full factorial design with high temperature, high CO2, and a combination of the two, 25 generations of selection. We're in the process of collecting epigenetic and population genomic data and sort of just getting our hands on that. So this should be coming together pretty soon. And again, I'd love to talk to anyone about this. We have some physiological data and fitness, everything across the generations. And we just started doing an entirely another species for this exact same experiment. So Carcia sonica is very closely related. It's much more geographically restricted and much less tolerant to low pH and high temperature. So I think there'll be this really nice comparison where we have sort of the species that is really likely to respond and be tolerant and resilient, and the species that's much more likely to be susceptible. And so that's sort of in progress now, and again, I can talk to you about how some of those results that we have. So with that, lots of people to thank, especially our COPAPOD collaborators at the University of Connecticut, Hans Dahm, Michael Finneguera, Hannes Baumann, and Jimmy DeMeo, my postdoc advisor Melissa Mespini, and NSF for funding. That, any questions? Yes? So the main thing we collected was fitness of egg production here on the on this axis, and hatching frequency. And the main effect we saw was that this is the full greenhouse. This is where we saw the effect. And I can talk to you more about this.